Okay, I'm Chi Ming Chi from Hong Kong U. So our program we call the LSCCB, Laboratory for Synthetic Chemistry and Chemical Biology. It's actually a tripartite collaboration with Imperial College and also Beijing University. So one of the missions hope to contribute is to make some contribution to make advanced cancer become a treatable chronic disease. Now, I think cancer, everyone knows, cancer is a life-threatening disease, but more terrible is the metastatic cancer, because tumor is really heterogeneous. Okay, sorry. And we have a lot of, cancer cell, more particularly with this kind of invasive cancer cell, cancer stem cell, and also some kind of immune inflammatory cells. The most current chemotherapy can only kill the primary cancer cells. It's more terrible, it is the fact that for some kind of treatment with the current chemotherapy, there will be some signal outgrowing to trigger the facilitate the growth of the metastatic cancer cell. There is some kind of cancer disease in the late stage. There's no therapy, like pancreatic cancer, and even with the lung cancer stage 4. So I take an example just to show you that the pancreatic cancer, now we did the experiments. This is the LSCCB, this is what we invented, one of the drug leads. So I mean the gemcitabine, and also that is used for the pancreatic cancer cell treatment. Basically, you can't cure the cancer. This is the clinical dosage. Also, we tested also with the cancer patient-derived organoid, cisplatin and gemcitabine at the clinical dosage, and actually could not it's not that effective. But still, this is used by the clinician, by the medical doctor to prescribe to the cancer patients. So this is our compound. So at this dosage, at 2 micromolar, it is pretty effective. So over the years, cancer, we know that the metastatic cancer, whether you like this or not, chemotherapy is indispensable. Particularly, actually, the platinum chemotherapy for lung cancer, 50% of the cancer patients at stage 4, they have to take the platinum chemotherapy. The reason why, because the current chemotherapy basically is non-specific and also has a series of side effects. There are a lot of cancer-promoting proteins. They're responsible for the angiogenesis, invasion, metastasis. None of, to be fair, in the clinical stage, there's no effective inhibitor developed to particularly to suppress this kind of cancers, this kind of cancer-promoting protein. So LSCCB developed proprietary anti-cancer compounds. So they have broad spectrum of activity superior to the clinical use chemotherapy, both to test it with the cancer patient-derived organoids. They targeted to the undruggable or oncogenic protein target. They are lacking effective inhibitors. They have strong anti-angiogenesis, anti-invasive effect, and also they can kill cancer stem cells. So I think I just give you some of the examples. So I give you the first example. You know that the platinum chemotherapy, cisplatin and carboplatin used for lung cancer treatment. Cisplatin has a pretty good efficacy, but it's a tolerable side effect. Carboplatin actually has better safety profile, but unfortunately the efficacy is not so effective. So we invented the so-called PTN from the LSCCB. So to make the story short, and actually it has much less systemic effect than the cisplatin. And also, of course, this is also, you could test it. It has the strong anti-angiogenesis, better than cisplatin, anti-migration, anti-invasion effect. We also, to show that it's really so better effective in the cancer patient-derived organoid, you compare the PTN, cisplatin at the 14.4, this is the clinical dosage, it's not that effective for the breast cancer. Now, the reason why for this is, we think the way is, the key form for the platinum chemotherapy is this unit, the platinum diamine unit. So we invent a so-called carrier, hopefully to recognize the biomolecule, the molecular target. And once you recognize the last like the DNA, we have the reversible integration and followed by very facile covalent binding. And we test this hypothesis, and actually is using the optical twister technique. So you can measure the binding force, and you can see the event, how this compound binds to the DNA. And for this one, we're also using the technique called nano-SIM technique to image, how the platinum goes inside the cell. The clinically used chemotherapy, the cisplatin, basically there's a very small amount of platinum in the whole cell, and that account why, and also it's pretty diffuse, why there's a lot of side effect. So this is our PTN compound. You have much more better accumulation. You can further modify this one. So I think this is something that we could, the next generation of platinum chemotherapy. The other one, I think that we say that is the so-called the gold bullet. For example, I take metastatic colon cancer as a model. This is the cancer patient-derived organoid. Clinical use 5-FUO and carboplatin, not that effective, but still used for the stage 4 colon cancer. So our compound actually is pretty effective. It's also very effective in the advanced stage lung cancer treatment. This is the cancer patient-derived organoid. I think what's more important, we know that this gold drug is pretty effective, but there might be the therapeutic window may be a little bit narrow. So we think that the best way is to do this antibody conjugate. I just showed this a very recent result. You think this is the clinical use of HER2 antibody and also this kind of linker. To make the story short, you take this, you inject this to the animal for 21 to 20 days, and then you stop the treatment. You can see here the tumor will not grow. I mean, this is, to me, is pretty remarkable. And I think at this dosage, the gold content, and actually, is just 0.35 milligram per kg, which is something we know that this is there's no side effect. Because we know the gold drug is, the side effect is only effective at about 1 to 2 microgram per kg. So I hope this is something that, and now we are working to develop this is for pancreatic cancer, because we know that this compound targets to some oncogenic protein like histoprotein 60 and also immunopeptides that could be effective for the pancreatic cancers. And finally, I'd like to make the story short by this. We also work on the TCM complex formula. This is very complicated, I think, but we need to provide the scientific basis. It has been used by the cancer patient anyhow. We saw that I want you to focus on this slide. This is for the pancreatic cancer. This is the clinical use of chemotherapy, cisplatin and gemcitabine. Basically, it's not effective at the dosage, but still, as I said, cancer patients still receive these treatments. You see the TCM1. This is pretty effective. I must say that this dosage is pretty safe. Cancer patients take this for months without any significant side effect. Of course, I mean, this is also not, we also should show that with some of the advanced technique, I mean, the biology, we show that this is reduced the metastasis for the pancreatic cancer cell to go into the liver and also to the lung. And of course, I think we would push this for clinical trial. I mean, so I think that was the future for this. We want for LACCB further develop to become an international chemistry innovation center, push this to the clinical trial. We have our imperial investigator want to have the Hong Kong set up the startup company. We encourage some of the new technique, DNA and co-library technique, nano-SIM technique. This is for the Chinese medicine, and also we hope to have more new opportunity and synthetic chemistry and also synthetic biology. This is particularly also for some of the PDL1, this kind of. Thank you very much.